Good morning, everyone. Welcome to today's Thyroid by MD's webinar. I am Camilo Gonzalez Velasquez, co-host of the Thyroid by MD's webinar series. Today, I am honored to host this session featuring a presentation by two experts in the field of pediatric thyroid tumors and genetic alterations. We're privileged to have here Dr. Janete Cerutti and Dr. Sharmila Sousa with us to discuss their recent publication on the genetic alterations landscape in pediatric thyroid tumors and differentiated thyroid cancer, a systematic review. We're also delighted to have Dr. Julio Ricarte-Filho join us as a discussant to provide valuable insights. Dr. Janete Cerutti is an associate professor of genetics and vice head of the genetic division at the Federal University of Sao Paulo. She directs the Laboratory of Genetic Basis in Thyroid Tumors and serves as president of the Latin American Thyroid Society since 2023. Dr. Cerutti has been recognized with numerous awards, including the Latz Young Researcher Award in 2001 and Senior Researcher Award in 2015. She was also named one of the 50 most influential women in science by the Open Box Science platform. Her prolific career includes publishing 115 peer-reviewed manuscripts and supervising over 84 students across various academic levels. Dr. Sharmila Sousa, a biomedical scientist with an impressive academical background that includes a master's in science in pharmaceutical medicine, a master's in research and PhD in medicine and endocrinology from the Escola Paulista de Medicina at the Universidad Federal de Sao Paulo, and master's in science in medicine science and society in King College in London. Dr. Sousa has an extensive experience in implementation in science projects and has been actively involved in developing gene panels and for the diagnosis and follow-up of thyroid conditions within the Brazilian public health system. She's held various esteemed positions, including roles at the King College in London and the World Health Organization and the Osvaldo Cruz Foundation School of Governance in Brasilia. Her work focuses on improving health technology, assessment processes, and policy development to inform decision-making in healthcare. Additionally, we have with us to drive discussion Dr. Julio Ricarte-Filho, a scientist in pediatric thyroid cancer translational research group at the Children's Hospital of Philadelphia. He completed a postdoctoral training in Dr. James Fagan Lab in Memorial Sloan Kettering Cancer Center in New York, and has a graduate studies at the University of Sao Paulo in Dr. Edna Kimura's lab. Dr. Ricarte-Filho research centers characterizing the genetic changes in pediatric thyroid cancer and understanding their mechanisms of action. We are very excited to hear from these experts today and look forward for a very interesting discussion. So without any further ado, I'll hand it over to Dr. Cerutti and Dr. Sousa to present their research. Thank you.
So can you hear me well?
Perfect. Well, yep.
Brilliant. So hello to everyone. Oh, just a second, please. Hello to everyone, and thank you so much for having us here today. We are truly honored by this invitation. These are our conflict of interest, as both Professor Janete and I have received funding from the Ministry of Health in Brazil to develop a feasibility study for the implementation of a genetic thyroid panel and a genomic classifier that we have been developing through various clinical analytical studies over the last 20 years, especially Professor Janete Cerutti. And myself, I also provide consultancy work for multilateral actors, as well as the federal government in Brazil, as previously mentioned by our colleague. Also, before we start, we would like to thank one of our co-authors, as these slides have been adapted from a presentation that she has done of our work last month at the Brazilian Thyroid Meeting here in Sao Paulo. So, both the American and the European Thyroid Associations have promoted special recommendations for the pediatric population, as they exhibit important clinical, molecular, and pathological differences in differentiated thyroid cancer when compared to adults, which obviously further prompts the development of context-specific pediatric guidelines at other global locations to fine-tune treatment according to age tiers. So in 2015, our group has identified the need to better explore sporadic cases, genetic data in detail. So we have been the first in 2016 to identify the prevalence of AGK-BRAF gene fusion in sporadic pediatric thyroid cancer, both by RT-PCR and Sanger, and also break-apart FISH confirmation. We have also reported in 2017 that fusions are the most prevalent genetic event in our population here in Brazil, especially those with less than 10 years of age, and that BRAF is a rare event in Brazilian older pediatric populations. We have then expanded our cohort investigation and identified AGK-BRAF fusion association with distant metastases in our younger patients. We have also outlined the case of a patient that has both AGK-BRAF and RET PDC3 fusions, further underlying that AGK-BRAF may be more associated with genetic instability due to telomeric aggregation. And we also outline different genetic profiles for our adult and pediatric cohorts in 2020, including also post-Fukushima and Chernobyl cases. So we have developed this systematic review to investigate the genetic landscape in pediatric thyroid tumors and also differentiated thyroid cancer, as we want to better understand the global and the Brazilian profiles for the genetic thyroid panel and genomic classifier that we are currently developing. Because we are looking to further detail child and adolescent genetic profiles per country and region, according to these two age tiers for thyroid adenoma, follicular and papillary thyroid carcinomas, and oncocytic thyroid carcinomas and adenomas, as well as the methods that have been deployed not only to identify, but also to confirm those genetic alterations for diagnosis and prognosis. So following PRISMA, we identified just over 3,500 publications at these databases that we outline here. After screening, we included 160 studies, which have been further categorized according to a series of critical quality appraisal tools, which comprised AMSTA2, as well as a series of Joanna Briggs Institute tools for each type of publications that we included, which were randomized control trials, cohort studies, case series and case reports, as well as text and opinion pieces. Exclusion reasons varied, but we want to outline that we did not include germline mutation and familial cases, as we are currently developing other systematic reviews, specifically for those tumor types. And we have also not considered microRNAs as diagnostic molecular tools, since their clinical pathological association with thyroid nodules and cancer are still being uncovered at global populations at present time. So our full genetic profiles evidence gap table per region and country is reported at supplementary table 1 for consultation. And we have outlined a series of methodological recommendations with clinical implications. So we start with the need for age stratification according to these age tiers that we have outlined here, or for authors to report individual patient age at diagnosis. Specific location data provision is also essential, we have found, as well as complete reporting of investigated and identified positive and negative results, as well as methods used for each specific genetic alterations. We did this because we identified a series of genetic evidence gaps that Professor Janete is going to explain now how they could be tackled by the establishment of new actionable information from research, development and implementation studies, that can enable adequate context-specific genomic tests and panels that are crucial for cost-effective precision oncology to be locally implemented as a short-term perspective at global locations.
So, good morning, everyone. It's a really pleasure to be here talking to you today. So, regarding the results, the global landscape of pediatric genetic alteration was created based on the calculation of the prevalence of each specific genetic alteration by subregion or stage, then country and region. Here in this map, you only outline the top 5 of each region. Additional genetic events identified in the 106 studies included in this review are available in supplementary data, as Sharmila just mentioned. You confirmed in this review that a common finding across the country was that in pediatric differentiated thyroid cancer, gene fusions were the most prevalent genetic alterations. Most commonly involving the red gene, with the rates varying significantly across the regions. The other most commonly identified fusion was involving an TRAC gene with different partners, especially with ETV6 in Brazil, Chernobyl, and Saudi Arabia affected cohorts. Although described in very few cohorts at very low prevalence, and therefore is not among the top 5 cited genetic alterations, AGK-BRAF is the third most common fusion in Chernobyl and South America affected cohorts. While ALK fusions are the third most frequent fusions in Europe, Fukushima, and North America. Regarding these needs, BRAF is uncommon in PTC patients with less than 10 years of age, being mainly found in children between the age of 10 and 18. Remarkable, in some regions or countries, BRAF V600E was not reported at all. Another SNP that was found was in Dicer gene. Although the precise role of Dicer is not fully understood, it emerged as one of the most common SNPs found in pediatric population in Asia, Middle East, and North America. Overall, regarding the RAS gene, mutations are found at very low rate in sporadic pediatric thyroid neoplasms and were associated with low-risk tumors. Therefore, since not contribute significantly to thyroid pediatric tumorigenesis. Regarding the genetic trends in clinical implication, although several factors could contribute to the difference in the prevalence of genetic alterations across the countries, it may be involved the use of not standardized methods and pointed out the need for global standardization. Therefore, outlining trends in thyroid genetics with clinical implications from a global landscape, those countries that use more comprehensive test methods, such as next-generation sequencing, were able to uncover new players in thyroid cancer. As mentioned, a common found between the countries was the presence of genetic fusions. You hear how highlighted the fusions involved RET and TRAC and BRAF genes, as they are not only important for the diagnosis in children, but also to the prognosis and treatment, as they were associated with aggressive phenotypes, lymph nodes involvement, and distant metastasis in children under the age of 10. We also outlined the need to establish transcriptional signatures of prevalent gene fusions similar to those reported in adult for BRAF and RAS genes. Regarding the Dicer SNPs, They were most found in neoplasias of follicular subtypes such as FTA, FTC, and follicular subtype of papillary thyroid carcinoma, including NIPT, which was found in Asia. Therefore, one of our recommendations was to report all genetic alterations, including those that co-occur with DICER for individual pediatric patients, because this will be key to identify genes that may contribute to disease progression. Regarding BRAF, they were rare in children under the age of 10 and RAS, a very common therefore, using BRAF and RAS scores are not appropriate for children. And this global epidemiological landscape is not only need to better understand the population's geographic and age variations, but also for detection of actionable targets such as tyrosine kinase inhibitors. If you do want to deploy such actionable targets for precision oncology purposes locally. In other words, if you do want to implement global precision oncology goals, you need to better understand each populational, geographic, and age variations for the detection of actionable targets such as tyrosine kinase inhibitors.
So I'll come back now to explain a few of the evidence gaps from our global landscape. So in Asia, with the exception of South Korea, we observed a lack of individual data on genetic alterations and clinical pathological features across several studies, which have prevented comparisons between different pediatric age tiers. In China, bridging the gap between evidence generation and its implementation therefore remains key, since preliminary reports of targeted therapy for progressive disease have shown encouraging results. In Europe, gaps underline the need for further studies in regions where pediatric population is fully understood at the molecular level. We have also identified that there's a functional analysis need to be made for specific variants and new fusions. And we need studies exploring genetic alterations across distinct foci to clarify differences between primary tumor and metastatic diseases. In the Middle East, there's need for larger and more comprehensive genomic characterization of sporadic papillary thyroid cancer to further determine genotype and phenotype correlations, because in fact, studies mainly investigated genetic alterations found in adult population. In North America, although a large series of next-generation sequencing panels have been implemented across several US states, new genetic alterations remain to be further underlined by in-depth methodological approaches, as many cohorts have been investigated, but individual data reporting remains to be implemented. In South America, retrospective follow-up studies have been deterring adequate follow-up. Sample size must be expanded beyond 3 Brazilian states and Colombia. And most prominently, detailed and transparent geographic differential analysis are needed before genetic tests or panels can be implemented across health systems. Finally, on gaps from Chernobyl and Fukushima affected cohorts, our global landscape outlined the need for studies to investigate differences in genetic alterations prevalence related to age, environmental and genetic factors, as well as techniques deployed, because individual social demographic data is also quite important and outlines the need for further mixed method studies on that front globally. We would like to thank all members of our group here cited as well as our partners and the funding agencies supporting us. Thank you once again for having us here today.
Thank you very much, Dr. Sousa and Cerutti, for your wonderful presentation and this very interesting presentation of your results. We will want to hand the mic over to Dr. Ricarte-Filho to see if he has a discussion to make around this topic.
So to start, I would like to thank Justin and all the Thank Foundation for the opportunity to discuss this very nice review from Dr. Cerutti's group, which is the Genetic Alterations Landscape in Pediatric Thyroid Tumours and or Differentiated Thyroid Cancer Systematic Review. I've known Dr. Cerutti for many years, ever since my undergrad studies in University of São Paulo, Brazil. And in fact, she was part of my PhD thesis committee. So it's a great pleasure to be here discussing her review. So I have no financial disclosures. And so what I would like to talk about, like in the next 15 minutes or so, is just go through like this review and from Dr. Cerutti's group and show a bit of the strengths and maybe some of the limitations and some of the main contributions that, in my opinion, it shows like from this review. So, and just a bit of a background is on pediatric thyroid cancer, we know that most of pediatric thyroid cancers comprise papillary thyroid cancers, which comprise about 90% of the cases. If compared to adults, they present with more invasive disease. They have larger tumors, a higher incidence of lymph node metastasis and distant metastasis, and greater risk for recurrence. And despite all this, these tumors have very, very favorable prognosis. They respond better to therapy than adults, only with like surgery and sometimes radioactive iodine. The most challenging cases are the ones with metastasis, like you have reduction in disease-specific survival when you have lymph node mets, and it's even more so when you have distant mets, in the way that if disease-specific mortality can get up to 10% in patients that have metastasis to the lung. And in terms of genetics, what we know is that most of these PTCs have genetic alterations activating the MAP kinase pathway, about 90% of them. And those are non-overlapping mutations of BRAF, RAS, and fusion oncogenes of RET and TRK1, 3, and ALK, among others. And all those alterations just lead to constitutive activation of the MAP kinase pathway. And we know a lot of this genomics of PTC from this study of the Cancer Genome Atlas from 10 years ago, looking at different molecular layers of characterization of PTCs, nearly 500 PTCs. This is from adults. And the most prevalent genetic alterations were BRAF mutations in 60% of the cases, RAS mutations in all three isoforms, 13%, as well as different kinase fusions that combined encompassed like 15% of them. So, and they showed like in this work that one of the major findings was the two-tier classification of PTC in BRAF-like and RAS-like, that based on the gene signature from these BRAF mutant tumors and RAS mutant tumors reflects better the clinical behavior of these tumors than pathological classification. And BRAF-like are usually associated with a higher MAP kinase output, lower differentiation score, whereas RAS-like are decreased MAP kinase output and higher differentiation score. As these tumors advance to poorly differentiated thyroid cancers, anaplastic thyroid cancers, they still have those initiating events with BRAF mutations, RAS mutations, but they also acquire some alterations that are associated with progression, just showing here third promoter mutation and p53 mutations and mutations of PI3 kinase AKT, activating the PI3 kinase pathway. These are common in poorly differentiated and even more common in anaplastic thyroid cancer, the most aggressive histology. And just to mention that third promoter and p53 are exceedingly rare in pediatric tumors. So those initiating events that I mentioned about the MAP kinase alterations, the pediatric tumors also have the same mutations, but there is a shift in prevalence. So what we see here is like in blue, that the prevalence of BRAF mutations is much higher in the adults if compared to the pediatric tumors. But when we look at the kinase fusions, what we see is that pediatric tumors have a much higher prevalence if compared to adults. And that is even more pronounced if you look at the pediatric tumors that are radiation exposed, as from this work here at post-Chernobyl samples. And not only they are more common in pediatric tumors, but they are more associated with invasive behavior. So these fusions are associated with tumors that have increased nodal metastasis, especially N1B, lateral lymph node metastasis, and distant metastasis to the lung, if compared to BRAF tumors or RAS mutant tumors. So we think that the fusion oncogenes are associated with more invasive behavior, and that's been shown in many different publications in pediatric tumors. So going back to the review, I think there is like different strengths from the review. It's a very comprehensive review of the literature, including the genetics of pediatric thyroid tumors. And after scanning like so many papers, still have like 160 papers that they included. And I think it's very neat, the fact that the geographical segregation and highlighting some of the differences in prevalence of genetic alterations from different regions. And also that supplementary file, which is a lot of work to put together, having all the histology, genetic alterations, and the methodology for every single study of the 160, which is very extensive as well. Just like not that many limitations, but it's sometimes it got like a bit confusing to me, like when we were talking about papillary thyroid cancers versus follicular thyroid cancers in during the review, like many times the differentiated thyroid cancers. And I'll show that like in a little bit with the example of figure 2. And also in terms of the mutations, many times when it's mentioned single nucleotide variants for DICER1, not clear if all those mutations are RNase 3B or they are loss of function mutations, because these are very different in terms of function of how they function in terms of microRNA dysregulation or tumor formation, and as well as the BRAF single nucleotide variants. I know the vast majority of them are V600E, but from the supplementary file, I could tell that there are some that are non-V600, and I'm not sure like if the prevalence included all of those or just the V600E. So going back to this figure 2, like where they nicely show this genetics of genetic landscape of pediatric thyroid tumors in different regions. And just wanted to mention some of the things like that. It was interesting to see the higher prevalence in the East Asian countries. And in fact, there are some reports associating that some of these countries may have a BRAF, a higher prevalence of BRAF in response to the high iodine intake found in these countries. Also, the increased prevalence of fusion oncogenes in the Chernobyl, which is expected, but it's a very high prevalence compared to any other geographical locations. And as for the other ones, it's just you see the regular increased prevalence of fusion kinase fusions and a lower prevalence of BRAF mutations. But also a couple of things really stood out here, which one is the AGK-BRAF at almost 19% in South America, the FGF receptor 3 single nucleotide variants at 15% here. And also, it was not included in this box, but one of the studies had a TERT promoter mutation prevalence of 27%, which is very high. So, and I just quickly mention each one of these studies here. So the one with the third promoter mutation is that work from China, and they show that this third mutation was presented in 13 of 48 pediatric papillary thyroid cancers. The work was done by Sanger sequencing, PCR Sanger sequencing, and but the traces are not presented in the manuscript. It is a very high prevalence because also all the other pediatric studies that were included in the review had either no third promoter mutations or a very low prevalence, which is like from 0 to 2%. And even in the adults where you see more of these mutations on TCJ project PTCs, you have 36 out of 384, so 9% prevalence is much lower than this. So you really have to take this with a grain of salt, like this high prevalence of those alterations in this manuscript. Another one is the FGF receptor 3 single nucleotide variant. This is a study from South Korea, and these mutations, they show that they found 6 mutations in 41 primary thyroid tumors, and they used a targeted panel. 4 of these were frame shift mutations, so it's loss of function alterations, and 3 were missense mutations with a variant of uncertain significance. So, in addition to that, these alterations were found to be concomitant with other driver alterations. So it's not clear what these mutations are doing there, but additional functional studies would be necessary to see if there is any function for these mutations, especially if they are found with other MAP kinase alterations. And lastly, like the AGK-BRAF fusion, which was in a study from Dr. Cerutti's group, they found a high prevalence of AGK-BRAF fusions in 15 out of the 80 pediatric PTCs. Although this is different because they do show all the traces for AGK-BRAF fusion in a second method of validation, which they used a fish in many of these samples that were positive. So it's more a question of why this population is showing such a high prevalence of AGK-BRAF fusions, that's an interesting question. And the authors also make some of remarks about like what they learned from this review, and many of the studies that were excluded because of some of these remarks. So many of the studies don't stratify results by age groups, like they just sometimes have a mix of samples from pediatric tumors, adult tumors, and it's not well like stratified and showed in the paper the age of these patients, and it's very hard to kind of segregate that. And it's important to do so because, as you can see from kinase fusion prevalence, if you look at kinase fusion in children from until 10 years old, like it's almost like all patients have a kinase fusion. In adolescents, you have a high prevalence of fusions, 45, 50%. And in adults, young adults, you have it's still like lower than pediatric tumors, but higher than the ones that are older. So it's very important to segregate and know the age of these patients as you report your findings, genetic findings. Remarked too, they say it's important to be very specific with location or the country. Sometimes there's like studies that are have samples from multiple locations, and it's important to detail like each one of these cases where they came from. And this becomes clear from like even figure 2 that they show in the paper, how like it changes from geographical location, one country to the other, continent to the other. So, and lastly, they say it's very important to really show, report each genetic alteration that was found, but also the ones that were not found. So basically show what methodological approach was used to detect. And I think like we can show the example we have from our studies that we just looked at fusion oncogenes or the genetics of pediatric tumors using 133 samples or cases, and we used two completely different panels, like one that was very comprehensive and we looked at six tumors with those, like targeted exome and 238 cancer genes, and over 600 fusions were screened. And other half of the samples, we used this mirror-informed panel that only looks at RET PTC1 and 3, BRAF V600E, RAS mutations, and PAX8 PPR gamma. And if you look at our results here, many of these cases that are indeterminate were screened by mirror-inform, which I just mentioned, just a screen for these alterations. So it's easy, like if you report that you're using mirror-inform, that you have the possibility to have other fusions here that were not screened, such as NTRK1, NTRK3, ALK, BRAF fusions, and RET fusions other than just the isoforms 1 and 3, as well as other fusions, MET fusion, and so on. And in fact, like by doing a more comprehensive analysis now with RNA sequencing, as you can see here, the BRAF alteration is close to 20%, and fusions are 33%. But after RNA sequencing, a more comprehensive study, what we see is BRAF is pretty much the same, but we were able to really increase those fusions that were not found in using the mirror-informed panel in a way that we go from 34% to 48% in detection of fusions, including some MTRKs and ALK and NTRK1, and some of the RET isoforms that were not found, could not be found with the mirror-informed panel. So just like emphasize some of the points that I wanted to about the review, but also in summary, like this is a very comprehensive review of the literature for the genetics of pediatric tumors, and it was really well performed with very strict and careful selection of articles to be included. It provided an overview of genetic landscape, and this is very important, especially for pediatric thyroid tumors, which is a very rare disease, and many of these studies had like very few samples, and so putting this all together is very important for us to make sense of the association of these genetic alterations, the real prevalence, and the association with the clinical features, and so we can make sense and really use it long-term for patient management. And so it really sheds light on the prevalence of certain mutations, different populations in geographic areas, and their association with clinical features, as I just mentioned. So with that, I would just like to thank and open this for discussion.
I want to thank everybody. Thank you. I want to thank everybody for their incredible presentations. I want to congratulate you and thank you for your results and for the advancements in the field that you are making. I have a couple of questions before I start with those. I would like to encourage the audience, if they have any questions, please send them through the Q&A function. And I'm sure, as we do often, we will get some questions via email from the people that watch this presentation after the fact on demand on YouTube. I wanted to echo what Dr. Sousa was saying. Do we need guidelines specific to this population, or do we need guidelines specific even to a specific geographical location? And I think you shed light on, when I saw that map and I saw the prevalence of the different fusions and mutations, that was interesting to me. But I always wonder and ponder if the differences that we find are based on studies that are made on big centers, academic centers mostly, and those are done normally in very diverse and big cities that might influence the results that we get. These are, like, if you go to New York, the population there is going to be more diverse than what I will find here in Monterrey, Mexico, where I'm standing right now. So are we finding differences in fusions, mutations based on where people stand? Is this a biological difference of a population? Or do you hypothesize that this might be based on different, exposures to certain different things, like the Fukushima and Chernobyl accidents the things that like that you might find? I don't know if anybody wants to take that question.
Yes.
Yes, good. Thank you. Sorry, because we are in the same room. I apologize. So yes, definitely. This is the main reason why we outlined these methodological recommendations and genetic trends that we identified. Mainly if we go to one of Julio's comments on the limitations that he outlined are the limitations from the literature itself. We mainly reported, especially to outline the findings and the evidence gaps and the need, especially in my last point, that if we do not look and try to develop different types of studies, maybe mixed method studies, so that we look for humanistic data that tackles, these environmental, the other social aspects that mainly are preventing us from identifying specific mutations or fusions in different populations across different regions. And sometimes even within the same state, if we have huge states like we do here in Brazil, and you also have in the US, and I'm sure for China, Russia, all those huge countries, it also happens. So this is quite important if we are aiming to identify these molecular biomarkers. Does that answer the question? Thank you.
Yes, thank you. It does. I
Can
wanted
I
to
just-
shift.
Can I
Yes,
just
please.
add one point, please? I think there is genetic diversity, but there is also environment factors that you should think about that may be important for this genetic variation found. But also the research and report bias. So there is no standardized method. So maybe if you use more specific methods to investigate the genetic alteration would help to identify the real prevalence of each genetic alteration in each region, country, or
Correct.
Okay.
Thank you. I wanted to ask you something. I'm going to change gears just a little bit. Regarding your systematic review methodology, you explained the criteria for including and excluding papers, and in the 160 papers, you excluded germline and familial cases. I know that you might not have final results, and it might be just jumping ahead of the horse a little bit here, but can you tell us what you hypothesized to find different in those patients with familial cases or germline mutations?
You mean which genetic alterations that are different in familial compared to pediatrics? Yes. So
Yeah.
you are doing some familial cases here, but most of the familias that you found are in the north part of Brazil, and they have several individuals affected by papillary carcinoma. Most of mutations we found was point mutations or loss of function mutations, like in DELs. In this case, there is some genes that were previously reported in the literature, but you still don't have any specific marker like red mutation for medullary thyroid carcinoma. So the mutations found in familial cancers are more difficult to find, more difficult to have a genetic test to implement in this case.
Thank you. You mentioned in your presentation and in your results the presence of AGK-BRAF gene mutation and its prevalence on pediatric thyroid cancer different from what we find in adult population. Can you elaborate a little bit on how this fusion particularly might impact the biological behavior of pediatric thyroid cancer and the aggressiveness that you state and we recognized in the introduction of your paper?
Yes. Well, you're doing functional analysis to more specifically understand and explain the biological behavior that we found in pediatric cancer. In our case, we found a very high correlation with distant metastasis, and most importantly, the AGK-BRAF fusion was found mainly in patients under the age of 10. So we were trying to better understand the behavior by doing functional analysis. I feel that very soon you have this data published, but I can now tell you that BRAF fusion behaviors more aggressively in the cell lines than the BRAF mutations and including the other fusions that we have tested in this functional analysis.
Awesome, thank you. I wanted to ask you another question. You mentioned in these studies that are using NGS where we find new players, as you stated, in the field. I wanted to ask maybe Julio, what do you think of DICER1, these AGK-BRAF mutations? How do they play a role, or can we make a genotype phenotype kind of relationship? Because these patients will grow and make that transition to adulthood, where we as adult endocrinologists or physicians that treat thyroid cancer will need to understand these fusions or different mutations that you are finding right now, and transition people into the different care that they will have further along their life.
Yeah, so we have a study with DICER mutations. And I mean, like, these mutations are much more prevalent in the pediatric tumors. They are very rare. Like, I think there's just 2 cases in the TCGA with DICER1 mutations. In pediatric, it's about like 10%. In some cases, like Dr. Cerutti showed, some cases are 20, some cohorts, 20% of DICER1 mutations. And those are like, it's very interesting. Like, these are associated with more indolent tumors. Usually they don't have lymph node metastasis or distant metastasis. And we have been studying also the mechanism of function of these mutations. When you have the RNase 3B domain mutation in DICER, that leads to microRNAs regulation. So you have loss of 5P microRNAs. Many of the microRNAs that are tumor suppressors, like LAT7, MIR30, are completely downregulated, and that leads to increased expression of some oncogenes, such as RAS, AGMJ2, and some genes that are important, and so and also regulate cell cycle. So we think it's also important, like although the vast majority are indolent tumors, sometimes you have, as reported by Nikiforov, some tumors with DICER mutations that are poorly differentiated as well. So we need to understand better the alterations that are involved in the progression of these tumors from like just being a DICER1 mutant indolent tumors, differentiated thyroid cancers, and these mutations can also be found in benign tumors. So what are like the different steps from benign to differentiated thyroid cancer, from differentiated thyroid cancer to poorly differentiated? And as for the AGK-BRAF mutations, I think like it's very hard to really get to a conclusion other than there's very few cases here and there. We just need to do things like Dr. Cerutti is doing, like putting it all together so we can make more sense of all the clinical features associated with these alterations, and functional studies that she's working on as well.
Correct. Well, this is very, very interesting. And I think I could go for another hour asking you guys questions. This is very interesting. I want to thank, in name of Thyro Brazil and all your inputs, all of you. I understand you're not all in Brazil right now, but I want to thank you and for pushing the field forward and giving us so incredible insights this morning. I want to thank you.
Yes.
Yes, good. Thank you. Sorry, because we are in the same room. I apologize. So yes, definitely. This is the main reason why we outlined these methodological recommendations and genetic trends that we identified. Mainly if we go to one of Julio's comments on the limitations that he outlined are the limitations from the literature itself. We mainly reported, especially to outline the findings and the evidence gaps and the need, especially on my last point, that if we do not look and try to develop different types of studies, maybe mixed method studies, so that we look for humanistic data that tackles, these environmental, the other social aspects that mainly are preventing us from identifying specific mutations or fusions in different populations across different regions. And sometimes even within the same state, if we have huge states like we do here in Brazil, and you also have in the US, and I'm sure for China, Russia, all those huge countries, it also happens. So this is quite important if we are aiming to identify these molecular biomarkers. Does that answer the question? Thank you.
Yes, thank you. It does. I wanted to shift.
Can I
Yes,
just
please.
add 1 point, please? I think there is genetic diversity, but there is also environment factors that you should think about that may be important for this genetic variation found. But also the research and report bias. So there is no standardized method. So maybe if you use more specific methods to investigate the genetic alteration would help to identify the real prevalence of each genetic alteration in each region, country, or
Correct.
Okay.
Thank you. I wanted to ask you something. I'm going to change gears just a little bit. Regarding your systematic review methodology, you explained the criteria for including and excluding papers, and in the 160 papers, you excluded germline and familial cases. I know that you might not have final results, and it might be just jumping ahead of the horse a little bit here, but can you tell us what you hypothesize to find different in those patients with familial cases or germline mutations?
You mean which genetic alterations that are different in familial compared to pediatrics? Yes. So
Yeah.
you are doing some familial cases here, but most of the familias that you found are in the north part of Brazil, and they have several individuals affected by papillary carcinoma. Most of mutations we found was point mutations or loss of function mutations, like in DELs. In this case, there is some genes that were previously reported in the literature, but you still don't have any specific marker like RET mutation for medullary carcinoma. So the mutations found in familial cancers are more difficult to find, more difficult to have a genetic test to implement in this case.
Thank you. You mentioned in your presentation and in your results the presence of AGK-BRAF gene mutation and its prevalence on pediatric thyroid cancer different from what we find in adult population. Can you elaborate a little bit on how this fusion particularly might impact the biological behavior of pediatric thyroid cancer and the aggressiveness that you state and we recognized in the introduction of your paper?
Yes. Well, you're doing functional analysis to more specifically understand and explain the biological behavior that you found in pediatric cancer. In our case, you found a very high correlation with distant metastasis, and most importantly, the AGK-BRAF mutation fusion was found mainly in patients under the age of 10. So you were trying to better understand the behavior by doing functional analysis. I feel that very soon you have this data published, but I can now tell you that BRAF fusion behaviors more aggressively in the cell lines than the BRAF mutations and including the other fusions that we have tested in this functional analysis.
Awesome, thank you. I wanted to ask you another question. You mentioned in these studies that are using NGS where we find new players, as you stated, in the field. I wanted to ask maybe Julio, what do you think? DICER1, these AGK-BRAF mutations, how do they play a role, or can we make a genotype phenotype kind of relationship? Because these patients will grow and make that transition to adulthood, where we as adult endocrinologists or physicians that treat thyroid cancer will need to understand these fusions or different mutations that you are finding right now, and transition people into the different care that they will have further along their life.
Yeah, so we have a study with DICER mutations. And I mean, like, these mutations are much more prevalent in the pediatric tumors. They are very rare. Like, I think there's just 2 cases in the TCGA with DICER1 mutations. In pediatric, it's about like 10%. In some cases, like Dr. Cerutti showed, some cases are 20, some cohorts, 20% of DICER1 mutations. And those are like, it's very interesting. Like, these are associated with more indolent tumors. Usually they don't have lymph node metastasis or distant metastasis. And we have been studying also the mechanism of function of these mutations. When you have the RNase 3B domain mutation in DICER, that leads to microRNAs regulation. So you have loss of 5P microRNAs. Many of the microRNAs that are tumor suppressors, like LAT7, MIR30, are completely downregulated, and that leads to increased expression of some oncogenes, such as RAS, AGMJ2, and some genes that are important, and so and also regulate cell cycle. So we think it's also important, like although the vast majority are indolent tumors, sometimes you have, as reported by Nikki Farov, some tumors with DICER mutations that are poorly differentiated as well. So we need to understand better the alterations that are involved in the progression of these tumors from like just being a DICER1 mutant indolent tumors, differentiated thyroid cancers, and these mutations can also be found in benign tumors. So what are like the different steps from benign to differentiated thyroid cancer, from differentiated thyroid cancer to poorly differentiated? And as for the AGK-BRAF mutations, I think like it's very hard to really get to a conclusion other than there's very few cases here and there. We just need to do things like Dr. Cerutti is doing, like putting it all together so we can make more sense of all the clinical features associated with these alterations, and functional studies that she's working on as well.
Correct. Well, this is very, very interesting. And I think I could go for another hour asking you guys questions. This is very interesting. I want to thank, in name of Tyro Brazil and all your inputs, all of you. I understand you're not all in Brazil right now, but I want to thank you and for pushing the field forward and giving us so incredible insights this morning. I want to thank you for carving out time and for being patient with the hiccup that we had for starting the webinar this morning. I want to give you back your Friday in any event, but I want to thank our audience for being here, and invite you back to be here next Friday to speak about medullary thyroid carcinoma and doubling times and tumor grade predictions. So that will be presented by Dr. Nigam, and with a beautiful discussion I know by Dr. Brandwayne Weber next Friday. So thank you all for being here. Hope you have a great weekend, and see you soon.